So today's lab is about phylogeny reconstruction, and that basically means the recreating of the actual evolutionary history of a group of organisms. And I set up this little video as an introduction to the whole concept. And what I want to do is to kind of go through with kind of a silly example, one that involves species that you're probably familiar with: the dog, the cat, mouse, and the rabbit. And so what I want to do here is to say, well, we all agree that at some point in the distant past, there was a common ancestor for dogs, cats, mice, and rabbits. And from that common ancestor, we had the splitting of the different lineages. At least 4 lineages emerge. Dogs, cats, mice, and rabbits all descend from that common ancestor in the past. One thing about this tree that makes it not an adequate, not an acceptable recreation of the history is that it's not adequately detailed. Yeah, we know that there's a common ancestor in the past, but the idea of having this polytomy, okay, basically when you have one lineage that splits off 4 ways, that's basically saying that there was one species that had 4 lineages that kind of all diverged from each other at the same time. The truth is that these lineages diverge at different points. We've got different branching points, and one of the rules that we have for phylogeny reconstruction is that we generally want to have one lineage splitting into 2. So polytomy is kind of a not allowed perspective on phylogeny reconstruction, and so we're going to be going with another model that requires us to have one lineage splitting into 2. So we're going to be getting rid of this picture and coming up with another alternative model which might actually depict a more detailed history. The one that I want to offer you here is one where you've got maybe a relatively recent common ancestor between dogs and cats. That's the one at the top. And maybe on the mouse-rabbit lineage, they share a common ancestor that's relatively recent compared to the more ancient common ancestor that's shared by all 4. You know, we still know that there was a common ancestor for all 4 species. What we're saying is that that ancient ancestor, the DCMR common ancestor, split apart into 2 lineages, one of which gave rise to the dog-cat common ancestor, still one species, and the other one giving rise to the mouse-rabbit common ancestor, still one species. And at some later time, the mouse-rabbit common ancestor diverged into the mouse lineage and the rabbit lineage, and not completely independently, the dog-cat common ancestor split into the caniforms and the feliforms. All right, so this is what we mean by a more detailed picture. There are other means by which we could sort of see this happening. I mean, this is just one possible history for the ancestry of these 4 lineages of mammals. Alternatively, we might have something that looks like this, where you've got a common ancestor of the mouse and rabbits together with the cats, and there's a dog, cat, mouse, rabbit common ancestor in the more distant past, further back. So way back here, this represents a different, we call it a different topology, a different branching pattern relative to the one that we had at first. And the breakdown of this is as follows. I mean, I've given you 2 different topologies so far, and I could recreate these 2 topologies right here. And you could say that in addition to these 2 topologies, or these 2 branching patterns, I could give you some others. For example, these are all variations on the same theme. Basically take the theme of topology number 2, and then we could say we'll keep the dogs separate and rearrange the mice and the rabbits and the cats and come up with a 3rd one. We rearrange further, come up with the 4th one, and then we could do further rearranging. There are actually 2 topologies here. Topology 1, this one over here, we might call this a bush because we've got one branch splitting into 2 and each of those 2 twigs splitting into smaller twigs. Whereas this other one, we can call this a ladder or a comb, in which you got one lineage splitting off and basically staying as a single lineage going off on its own direction, the dog lineage, and then we've got another one that splits off and stays on its own. And the shape of these 2 kinds of trees are the bush, respectively, on the left, and the ladder on the right. Okay. And these are all different trees. They're all qualitatively different trees. Okay. And the first thing that I want to point out here is that it's not that every tree you can possibly draw out here is going to be different. There are some trees that might be synonymous. Okay. So I have an example here, number 8, layer 8 for me. Here are 2 trees that if you look at them really carefully, they're actually synonymous with one of the trees that we have in the top row. Can you tell which one it is? Yeah, you might want to pause the video here to see if you can tell which one of the top 6 trees is exactly the same as what both of these trees are saying down below. And if you have the answer, great. Here's the answer. Okay, it's tree number 5. Tree number 5, if you look at it, they both have dogs and mice sharing relatively recent common ancestries, and cats and rabbits sharing common ancestors. And that same topology is reflected here. When it shouldn't be too difficult for you to see that all 3 of these 3 trees are saying exactly the same thing. Okay. So what we want to do is we want to be able to distinguish between the trees that really do say different things, and we can get rid of these 2 pictures and start filling in with other trees. And so what I've done is I've put together what I think is a pretty comprehensive list of all the trees that are possible. And here you're actually seeing a total of looks like 15 trees that all say qualitatively different things. And another feature about these trees is that one of them has to be the truth. Okay? So out of 15 trees, do they all have an equal likelihood of being true? And it's kind of like a random shot in the dark to say one of these is actually the history of our mammals? Well, no. I mean, I think when I do this in class, almost every time we get most of the class agreeing that one of these trees is actually far more likely to being the truth relative to all the other ones. In other words, we have in our minds this mechanism of doing phylogeny reconstruction without really knowing it. This is kind of like an intuitive thing. And so if I ask you to decide which one of the 15 trees is the best, are you ready to move on to the next step? You can pause it here. And when you're ready to start again, just unpause the videotape or the video, and we'll give you the right answer. So the right answer, at least not necessarily the right answer, but the answer that most people generally come up with is this one. All right. So yeah, tree number 1 is generally accepted as being the more likely of all the trees that we have on the rest of the board. Okay, can you see why that is? Well, here's where I usually ask students to describe for me why they think that one tree is better than all the others. And the answer is usually pretty consistent. Okay, and so we're going to go back up to the top and go and get rid of this tree. We don't like that one. And get back to the tree that we did like. That's the one that we had. So this was a tree that we liked. And I guess the question is, well, what is it about this tree? Okay, I'll make this more centered. What is it about this tree that makes it better? Why do we have such strong conviction that this tree is far more likely of being the true one relative to all the others? Well, it boils down to something that's very simple. I think you could say, well, it's based on similarities and differences. Dogs and cats share a lot of similarities. These things that dogs and cats share are things like dentition, the way the teeth are, the shape of the skull, the shape of the bones, the characteristics of the fur. Mice and rabbits are also very similar. They've got similarities, and that's why we put them together on the same part of the tree. At the same time, there are differences. There are differences that say, well, mice and rabbits don't have the characteristics that we find in dogs and cats. So here's where we get to the evolutionary part of the story, which is to understand the causes of similarities and differences. The reason why things are similar is because of the phenomenon that we identify in evolutionary biology as homology. Dogs and cats are similar because they share characteristics that were there in that dog-cat common ancestor, the DC common ancestor, that were simply inherited unchanged from that ancestor. Okay. Mice and rabbits similarly share similarities of dentition, of skull pattern, of skull bone morphology, of skeletal morphology, of physiology, of digestive physiology. There are many things that make rabbits and mice morphologically very similar, and those characteristics were largely, presumably, inherited unchanged from the mouse-rabbit common ancestor. Okay. Together with that comes the other part of the story, which is the causes of differences. And this is the other side of the story, which is to say that the reason why dogs and cats and mice and rabbits are different from each other, at least the mice and the rabbits are different from the dogs and the cats, is because they do not represent part of the clade. In other words, a mouse is different from the dog and the cat because it's not part of the clade or family that descend from the dog-cat common ancestor. And not being part of that clade, the mouse never got the characteristics that are shared by dogs and cats. So there's a homologous character state that is there in dogs and cats that we don't find in mice and rabbits. And there are homologous character states that are present in mice and rabbits that you don't find in dogs and cats because, again, a cat is not part of the clade that is descending from the mouse-rabbit common ancestor. And here we come to a really important concept. Here's another buzzword for you, and I'll spell it out for you right here. This is the word synapomorphy. Which comes from two parts. Syn means together, and apos means separate. And a synapomorphy synapomorphies are really important for a discussion in Bio 202 because these are the characteristics, these are the homologous character states that are shared by all the members of a clade. The dogs and the cats share synapomorphies of dentition, of skeletal morphology that are not found elsewhere. So in other words, these are the characteristics that unite dogs and cats within the same clade that also separate them from other animals that are kind of closely related that do not belong to the same clade. Okay. So this is a term that we use over and over and over again in Biology 202. And this is a great place for me to introduce the concept. All right. Now, second part of the video. This comes to us from the idea that, well, in our traditional view of things, in a traditional taxonomy, we're looking primarily at taxonomies that were put together by morphological systematists, people that were looking at the bone structure, at the measurements that you find on animals and plants. In the last 25 years or so, we have access to another kind of character state, set of characteristics that are entirely molecular. And so the genes, the nucleotide sequence in a particular gene that you can isolate from different species, represents a wealth of taxonomic information that can be used to recreate phylogenies. And this is kind of like what we're doing with today's laboratory. With today's lab, we'll be using a program, Genius, to download sequences from different species, 4, 5, 10 different species, however many you have in your data set. And we're going to be using those sequences to reconstruct the evolutionary histories between those organisms. So in other words, we're not looking at animals with teeth and fur and behaviors that are different or similar to each other. We're looking at genes in which the nucleotides might be the same or different, presuming that we're talking about homologous genes, right? So you need to make sure that you're talking about genes that really do mean the same thing in two different species, right? So what we're going to be doing here, we'll be comparing the gene sequences, the nucleotide sequences between the same gene that's been isolated from different species. And here I've given you species 1, 2, 3, and 4. And we don't even need to know what they are. We're just going to be using the data that are there to decipher which organisms are more closely related based, again, on similarities. The more similarities we have between two species, the more likely they are to share a very recent common ancestor. The greater the number of differences, the less likely it becomes that they share a recent common ancestor. Their ancestry must be pushed further back into the past. So in this case, we've got 4 sequences of 11 bases that have been isolated from the 4 different species, and we're going to be looking at comparisons between the 2 different species and as it comes to the molecular characteristics. Okay, so let's start by comparing species 1 and species 2. Right? And when we do that, we'll look down the entire sequence. We'll say that there's this one difference between the 2 sequences at position 5. Now when you do this, you're going to be looking at a lot more than 11 bases. Okay, but if we do this for our 11 base sequence, we've got a total of 1 difference between between them. Okay, and we could do the same for every pairwise comparison. So we've got species 1 and species 2 with 1 difference out of 11, species 3 and species 1. If we compare the species 1 and species 3, we can see there's a difference between A and C at base number 2, and then at base number 5, we've got a difference between T and C, and base number looks like 9, we've got a difference between G and A. So there are actually 4 differences that we can find between 1 and 3. And you could look at the differences that compare 1 and 4, and between 1 and 4, we've got 6 differences. And then we do the same thing for the comparison between 2 and 3. We find the 3 differences between 2 and 4. there are 5 differences out of 11, then the last one that we want to do here is to compare 3 and 4. And when we do that, we find that by comparing 3 and 4, there are 2 bases that are different between those 2 sequences. Okay, and when we put this together, it looks to me like we've got 2 pairs of species, 1 and 2, that are very similar to each other, 3 and 4 that are also very similar to each other, and the rest are more distant. 2 and 3 are pretty similar, but we're going to work with the idea that there's strong evidence that align species 1 and species 2 closely together on the tree of life, and 2 differences separate 3 and 4, and we might use this information to put together a tree that connect the 4 species. So here you would be saying species 1 and 2, again on the basis of there being only one difference between species 1 and 2, they belong together on their own twig of the tree of life. And species 3 and 4, based on their very close similarity, there are only 2 differences out of 11, they belong together on their own little twig. And then those 2 twigs are going to be similar, are going to be related to each other in this way out here because they have to share a common ancestor. The differences that we find between either 1 and 3 and 1 and 4, 2 and 3, 2 and 4, those differences are all greater, and from that we infer that there's going to be a more distant common ancestor. So this might have been the results we got for dogs and cats and mice and rabbits. Okay. And this would be one way that we would we'd use this to confirm our previous cladogram or previous phylogeny by saying the molecular data are supporting the idea that dogs and cats are closely related, mice and rabbits are closely related, and the ancestor connecting the dog-cat ancestor and the mouse-rabbit common ancestor is far more removed in time. It's more ancient. All right. Oh, and there's one more thing. Okay. If you look at, if there's another form of inference that we can make from these cladograms or phylogenies, and that's to use information that's embedded within the phylogeny to determine answers like this. For example, if you look at this dendrogram or cladogram, you notice that between species 1 and species 2, there's a difference at this 5th base. C or T, and you could ask the question, if you come back to this ancestor right there, is it going to be T or is it going to be C? I mean, was it T and then this lineage, the lineage that led to 1 changed to C, or is it was that ancestral form a C and then it's this other lineage changed to T? How can you tell between those two? Well, as it turns out, we have information, we have data embedded within the table. We can look down here at the other species in the dendrogram and say, oh, at the 5th position over here, both rabbits and mice have a T, and at that point we could say, oh, it probably is this. This was a T at the ancestral bit, because we know that because this information here is leading us to the conclusion that that was a T. In other words, we're basically getting information about this ancestor by using the data that's coming from the other limb of the tree of life. Okay, and so the way that this information is it's important to us because it's at some point it's worthwhile for us to know the character state of that branching point. We can presume that everywhere along this lineage it was T, T, T throughout this whole time, and it was only along this lineage here that we had to switch from T to C. Okay, it's another bit of detail that's being added to our understanding of the history of these animals. Okay. Now, when we look at the entire cladogram, though, if we look at the entire cladogram, we realize that there are places where the same question can be asked about the most ancient common ancestor. So, for example, if we look at the 2nd base, we notice that both dogs and cats have C's there. So we can presume that this ancestor right here, the dog-cat common ancestor, had a C at this position, and we have a T at this position, or sorry, an A at this position. Because we have an A at both for sequences 3 and 4, we can infer that this ancestor here had the character state A at that 2nd base position. Okay. And then the question goes, which was the character state? Was the ancestral point here A, or was it C? And you can't answer that question from this tree because we don't have any reference points. See, that's kind of like the big question. So in order to answer that question, we like to include, with any kind of phylogeny reconstruction, we like to include what's called an outgroup. Okay? And in this case, the outgroup will be something that's not related to cats or dogs or mice or rabbits. It's got to be something that's outside of the clade that includes all of those four species. So you might choose maybe an elephant. An elephant would be a good example. And so if you take the elephant genome and you find that the elephant genome has got the sequence A, C, C, etc., you might be able to use this information to say, okay, well, the outgroup has the character state of A, and we could use this to infer that this ancestral state for our original ancestor, the dog, cat, mouse, and mouse rabbit ancestor, had the character state of adenine at the second base. Okay, so in your analyses, I would like you to include an outgroup. You can consult with me to figure out what a good outgroup is. The important criteria for a good outgroup are two, really. The two criteria are one, that it cannot be part of the clade. You cannot choose as an outgroup an organism that belongs within the clade that you're trying to decipher. Okay, so if you're looking at the different kinds of dogs, if your phylogeny reconstruction is about all different kinds of wild dogs, Canis familiaris would not be a good outgroup because it's actually derived from the wolves, which are part of your study. And so Canis familiaris is an in-group, and therefore it would not be a very good choice to force on the outside. The other characteristic that is important is that you choose an outgroup that's outside the clade, but not too far outside. So if you're studying different species of dogs, you might want to choose maybe a bear as an outgroup, which is pretty closely aligned with the caniform clade. You would not want to choose jellyfish as the outgroup because that's so far removed it's not going to be giving you that much good information about the immediate common ancestor for all the dogs, right? So those are the tips that I wanted to give you, and I think the rest of the information is spelled out in the other video that I've already put online.